We have deferrals that are taking place this week, and I was very busy preparing those as well, so it's going to be on the Thursday. I will be looking into your concerns for sure. Whoever has any issue, feel free to come to office hours and we will discuss your concerns. Okay, now the assignment number 2 is almost done. I'm just waiting the final confirmation from the TAs so that we could publish the grades. Overall, I hope you have done well in this one too. And I just want to share with you some statistics. Like in the assignment number 1, although there were lots of complaints, the average of the grades were between A and A plus and A minus. So the most of the class has really got good grades. There were a few grades not okay. And as I said, I will work on adding a quiz or so to compensate. I just want to know, you guys hear me where I'm talking? Someone just say, you hear me?
Yes.
Oh,
We can
okay.
hear you.
Okay, good. Because it looked I'm silent. The other thing is, I'm not sure if you noticed that some of your midterm grades were adjusted because we went through the exam. Of course, the multiple select questions, it was very clearly stated there is more than one answer for only 5 questions. The rest, they had only one answer. And there were the fill in the blanks. And those, no matter how I put the results in the bright space, if there was a comma or a point that you didn't write, then the bright space is going to consider your result wrong. So to be fair for you, we went and we corrected the fill in the blanks. And that's why if you have seen in the last 48 hours some changes, of course, an addition in your grades, this means the system had declared some of your results wrong, but the meaning of what you wrote was correct, so we provided the grade. So this is just to answer a general question that everybody was saying in the email. Probably the fill in the blanks are not corrected automatically with high precision, so we went and corrected those manually. Could you please go to your bright space, and I would like to have a reflection from you about the mid for 2 reasons. First of all, I would like to know how complex is this course and the exam was, and I would like to get some suggestions from you in order to make the final exam better and easier. So please go to your bright space. And if you go to the content, there should be a midterm evaluation feedback. Please go to that one.
Professor,
Mm-hmm.
I was wondering if you could, if you're able to post the right answers for the quizzes.
As I said, we still have the deferrals, and I will look into that after that.
Okay, thank you.
Okay. Okay, can you see my screen?
Yes, we can.
Okay, so let's go to the WooClap, and I want to just put this screen here. So in case you don't have the bright space, you can go to the event. That's the code. You can see the code here. Again, you are anonymous, so I don't know who is answering what. So feel free to quickly answer these questions because we don't want to take too much time on answering. These are very straightforward questions. You don't have to think about the science behind it. So the majority didn't get the grade they expected. Okay. Next. So the midterm was easy, difficult, or fair? Mhm. Uh, Kyla, I haven't got into that yet because I'll do this when all the class has done. Adora, what do you have? Adora, can you, or you write in the chat? Okay. Now, if the midterm was difficult, I want to know why. So, were the questions in the exam covered in the slides? Adora, please write your question in the chat. Okay, so I am 100% sure every single question came from slides. So for those 8 people who said no, you can argue me by showing me one of the questions that I literally went to each slide and made the questions. Now, I also included some of the exact questions that I made in the book lab. So that tells you, whenever I tell you that there's a question that comes from a certain slide, you have to trust me that I will really ask that question. At least five of the questions or 6 of the questions, I exactly copy pasted from the WOOC lab that we discussed during the classes. But that's good. I want to take advantage of this review because I want to make things easier for you. If there was some misunderstanding, we want to kind of remove any gaps. So when you were preparing for the exam, did you really go visit the resources? Like you watched the YouTube, went to the website, went to the books, sometimes pages, because this is also essential. Many times in the slide, I cannot include everything. And if I include everything, it will be so crowded. And if I include everything, it will be so much information that I can't cover during the class. So that's why there are additional resources most of the time added to the slides, like links or resources in the content. So, okay. Okay. I still see that some people are not answering. I mean, these questions should be easy, guys. Please go ahead. I wanna just get a flavor how to make things better for you. Okay. The other question I want to know is, how do you compare this course overall to other courses? It's like, do you think it's good, it's bad, you expected that it's easier than this? Mhm. There's still 32. Please, I need more. Mhm. Okay, so the majority of you think that this course is worse than other courses. Okay. So for those who think it's bad, in the next session, I will have another WOOC lab to give you more space to explain why you think it's bad. And I'm not saying that I'm the perfect professor. I'm trying to do my best. I'm trying to make things as easy as possible for you, but at the same time, I'm maintaining a particular level in the course. Like, you still have to know something at the end of the course. You can't simply come to a course and leave and you don't retain any information. And maybe next year you will understand what I'm saying, because many of you next year will be doing honors projects or trying hardly to get to a lab to perform any research in clinical wise or in like basic research, and then you will understand how important it is to know these details. But thank you very much for being honest, because that's important for me to know that the course is not meeting your expectations, or at least the majority of people who are here. And it's like a little bit tough for me that only one person thinks that this is a good course out of 100 something, 43 present today. So how do you compare my performance now? Forget about the course itself. Do you think it is fair? Do you think it's bad? And again, you are anonymous. I don't know who's saying what, because I want to know what should I do to make this course better. And it could be at my level that there's something wrong. I want to really look into considering my performance, my way in presentation, my way in approaching your questions, follow-up, feedback, emails, TA problems, corrections, all that. I really respect whatever you say. It's just I want to know how to make things better from my personal perspective as a professor dealing with the overall. So there are still 5 people. Please, if you can answer, because I want to give you some space to express yourself. Okay, so let us go to the votes. So, and this whole club can be answered anytime. So what would you suggest to make the next exam better? Okay, so please feel free to give me suggestions so that I take those into consideration. No multiple select, open book. No multi-select or fill in the blanks. More practice. No fill in the blanks. Only multiple. Okay. Less or no multiples. Oh, you guys don't like multiple select. Okay. Okay. Practice. Okay. Feel free to add as much as you want information here because I will review this later on. Just for practice questions, I literally, guys, every other class I have WOO clubs. So do you think you are not getting enough practice? Mock exam. Okay, that's a good thing. Mock exam. Less proctoring. Okay. Okay, less proctoring. Less proctoring is something that would look into consider topic. Okay. No fill in the blanks. Okay, so I can get at least 2 or 3 major things that you didn't like. Clearer. Oh, wait. What? Okay. Guys, last year in the pathophysiology, I used to grade the slides. So I used to tell them which slides are going to be included in the class. Like I say code 0, 1, 2. 0 means there's no chance that will be a question, but it's needed to understand. 1, there's a possibility of a question, and 2, there's a question coming from that slide. So probably we should do this in the final exam. Okay, sounds good. So let's move to the next. So what do you suggest me to change in the course? What do you don't like about the process? Do you prefer having, for example, a book that you read from all the time instead of just the slides? Do you prefer having more visuals? Make the assignments easier to follow. No book. Provide with club question answer after class. By the way, the Woo club are already in the videos. So right now we are having them in the video. They are recorded. So you can easily come anytime and watch the question answers. And we were all showing the answers. Slides are a bit messy, provide better explanation, text associated with each lecture. Okay. And so now let me just quickly answer some of the so there's this comment I'm trying to hire a city boy JJ as a TA. Okay. Can you post the slides a day before? This depends on how busy I am. I will try. Easier questions. Slides are messy. Clearer slides the way the information. No textbook. Okay. Easier exams. You are a great prof. Thank you. More straightforward assignment questions. Like, okay. For this particular comment, more straightforward assignment question. The second assignment was absolutely straightforward. There's no way you can tell me that the second assignment was not straightforward because we kind of went through one whole lecture, how to get a gene, how you get a sequence, how you get a main transcript. Then after I gave you the assignment. I went for another 20 minutes and I showed you one gene, how you get all the information. Literally, you needed less than an hour to finish that assignment. For the second assignment, I needed more info that, okay. For the second assignment, I needed more info on how to make a primer. The slides and the videos were unclear. Okay. Well, I figured out, so from this particular question, I figured out that when I went back to my video and I was showing you how to test the primers, it looked like that window, it didn't open in the video. Like you were not seeing that window. And I was really shocked that nobody told me that we don't see anything. We still see the same screen. So too bad from your end and from my end, because I reviewed the video later. Okay, so very good. Discussing the chirality of the CDNA. Exactly. So this is what was not shown. So I was clicking on checking on the complementarity of the DNA. Then I saw that when I click on that window, it doesn't show on the video. So it was not taken. So, but anyway, feel free, guys, to stop me when I'm presenting and I'm saying something and you don't see it. I can't see what your screen is showing, right? And yes, thanks. One of your colleagues said about the pointer. It makes things better. And the summary slide at the end of the class. Most of the time, I wouldn't have the time to do that because, as you can see, we spend a lot of time discussing in the beginning. But the learning objectives is more or less in the beginning of each slide. It's a very good idea. PhD. Okay, labs. By the way, guys, I discussed with the department if you can have any training. Then I know there is a program. I'm going to send you an email about it, about 60 hours where you can do research in certain labs. Previously, it was paid, where you get $1,000 for this 60 hours. But unfortunately, the university discontinued paying. So you will be doing that for free, but then you still have to find a lab where you can do most of these techniques that we are discussing. Okay, so thank you very much for your honest opinion. I will look into these and I will come back to you next week after discussing these, summarizing this information. As for now, let's just move on to the, okay, let's move on to the lecture of today. And you recall that we still have 2 lectures in the DNA applications, but since we had, okay, so let's minimize this. Please, if you want to say something, I can't see because I'm presenting now and it will be difficult to catch chat. Either write a note or come back by the end of the lecture, or unmute yourself if you have something to say.
Professor, just to make sure, is a final exam non-cumulative?
Accumulative in which sense?
Like the contents on the midterm, is it going to be revisited in the finals?
No, no. The content is from this lecture up.
Okay, thank you.
Sure. Okay, so as for today, we will discuss the applications of the DNA sequencing and the recombinant DNA technology, and how do we apply that in genomics. We will also try to make a roadmap for the end of the lectures by discussing the transcriptomics and the proteomics. So it will be just a couple of slides about those, because there will be dedicated lectures for studying the RNA analysis, gene expression, and the protein analysis and proteomics. So again, the office hours are still set 12 to 1 on Wednesday. You can always use the unified link, and the recordings are always available on Zoom and on the YouTube in case you have issues with the cloud. Bear in mind that on Zoom, you can't keep the recordings forever. Now, so this is the roadmap so far. We discussed in details about amplification of DNA, sequencing procedures. We discussed how you can identify a gene, how you look for a gene on the Ensemble website and NCBI. We discussed the restriction enzymes, their applications in recombinant DNA technology. We discussed some types of the plasmids, not all of them, and how we can deliver the plasmids to the bacteria and to the eukaryotic cells. So overall, this is what you can cover in a lab. Some of what we will discuss today is more about knowledge than practice that you can do. In other words, genomics is not something that a lab can do. It needs many big labs to do transcriptomics or genomics. And generating transgenic animals is also something that we need a core, or you need like really university to do that. So researchers by their own, they don't really do all these transgenic animals. They have the idea behind why to make a transgenic animal and why they want to study this transgenic animal, but they don't do it by hand. And therefore, this lecture, probably it will give you just a background. And again, before the final, let's do this coding for the slides so that I can eliminate slides, that I will not ask you questions in the exam, but it's important for you to be a reference that one day you needed to understand something about genomics, transcriptomics. You can come back and you can get into these details. So the major points we will discuss today is the Human Genome Project. And the Human Genome Project is the process of obtaining the sequence of all the chromosomes of the human genome, right? Without knowing all the details on this slide, starting from extraction of DNA, detection, quality check, amplification, sequencing, knowing how to annotate the sequences, how to put them in databases, how to obtain them from databases, and restriction enzymes. In addition to the discovery of plasmids and other vectors, like we speak about plasmid as a vector because it's very well used, and other plasmids, or like BAC and YAC, which is the bacterial artificial chromosome and yeast artificial chromosome. Without all these, genomics and transcriptomics and transgenic animal were not possible. So now we will move from having that simple application of studying individual genes by knowing all these techniques that we discussed before, and notions and plasmids and all that, into comparing the genomes of human to mouse, for example, or knowing all the sequence of the human genome, annotations, knowing where are those genes located, and how they are carried over and all that. So now we will just discuss how we got into getting the whole genome sequence. And this is a project that, of course, it is part of many other big projects that are running right now in terms of research, because the building block for that is really the DNA sequencing technologies. Because with the emerging of novel sequencing technologies that elaborated and make the sequencing much quicker and much more efficient, then all these fields start to propagate very quickly. So with DNA sequencing, we could know the structure of the genes, the function of the genes, we could know the evolution of those genes, how they changed, and we could map those genes back to the chromosomes, and we could edit these genes to make, for example, as we discussed in the previous lectures about CRISPR and making transgenic animals to study the role of a gene in vivo. Of course, the epigenomics, which covers more the epigenetic marks on the DNA itself, which could affect a gene transcription by turning a gene on or turning a gene off. This is another whole different domain of study. And transcriptomics, where if we know the genes, then most probably these genes will have a function, and that function is not going to be really played by the gene at the DNA level only, because it has to be transcribed into the RNA. And those RNA, they could play a part of the function. They could relay the message to make a protein, and the proteins are, in principle, the functional units. So transcriptomics deal with the complete set of the RNA that has been transcribed. Now, in terms of transcriptomics, there will be one whole lecture about discussing the different types of RNA and how we analyze the RNAs. Mainly, transcriptomics focus on the messenger RNA, but now there are new RNA classes that are coming into the image, and particularly the long non-coding RNA, because now we know that most of the genome doesn't really get transcribed into messenger RNA, but into probably long non-coding RNA. So this will be a whole new lecture in the next few weeks. And finally, we will discuss the interactomes or the proteomics, where we will discuss first the whole protein that is being generated from a genome or a genetic material, and then the interaction between the protein and the DNA, because in principle, when you do transcription, there is a protein that binds to the DNA, which is like RNA polymerase. It's an enzyme. It's a protein. So this will be also another lecture about how RNA and how DNA and protein interact. Okay. I will not cover the epigenomics because I doubt that any of you will need it if you go tomorrow for a lab. But I will give you some resources as well. So, for example, this is one of the slides that you probably don't have to retain information except only a couple of sentences. And I really want from now on to clarify for you what makes a part of the slide important and what is not. So in terms of genomics, you would like to understand where a gene is located. So if you look at this image here, I don't expect you to know the details on this chromosome, but it can tell you, if you look at the zoom, that there's this gene, for example, actinin. This gene is located between two other unknown genes. So when we want to start to discuss the genomics and understand how the genetic work, there are multiple aspects you have to take. First is, if we are studying the organization of the genes, the content in terms of the sequence and then the function. So that is overall the genomic world, if I want to say. But if you want to look into that from a structural point of view, then you are in principle looking for the sequence which makes that gene. But then that sequence is physically located somewhere on a chromosome. We have 23 chromosomes, so that sequence is physically located somewhere on a chromosome. And from here came two main concepts. The genetic map, which is mainly linked to where is this gene located, or in other words, it's a linkage of the gene to other boundaries or surrounding genes. So it will be very easy for you to understand that, for example, actinin, if it's transferred from one generation to another, it's most probably carrying the other two genes. Most probably the other two genes are going to be carried over. And that's why the genetic maps are mainly linked to the process of having, if there are two markers that are close on a chromosome, if one of the markers is transmitted, then most probably the second marker is going to be transmitted. So this is one of the concepts how you look at the genomics. The second concept would be the physical map. And the physical map, it's in other words, the exact sequence. And this is, if you remember, we discussed in the assignment, when I ask you, for example, go and find me what is the genomic DNA of the IGF gene. So you directly went, oh, it's on chromosome number 12, and it is located between this base pair and that base pair. So in principle, you gave me the physical map where the gene is located, and you gave me the sequence of that gene. So these are two different concepts, okay? Now, if we want to study a gene, then we can study that in different ways. And up till now, everything I described for you was to understand how a gene works. So in other words, I described big, complicated aspects at the level of one gene, but in the next couple of slides, I will be applying those to the whole genome, and you will see how that you already have the basics. It will be very easy for you to understand how the genome project proceeded. So first, let me remind you that we discussed the genomic library, and we said that if we want to create, if we want to study a gene that we never understood before, we never had any information about before, we take DNA, and then we make a library from the DNA. In other words, I will first have the first difficulty to create that library because there's a lot of DNA in the cell, around 3.2 billion base pairs. So what can I do in order to make a library? With the discovery of the restriction enzymes, we know that the restriction enzymes cut at different sites in the genome. So when I take all these 23 chromosomes, then there will be random restriction sites for a particular restriction enzyme. As you can see, they are labeled in red arrows here. So based on what vectors we're going to use after isolating that DNA, then we will cut or digest the DNA with a particular restriction enzyme. And in this example here, this is the cosmid, or it's the viral vector. So if we cut the DNA up to 35 to 44 base pairs fragments, then we will have 350 clones that are going to cosmids. And in this situation, you can cover almost 99 point something of the whole genome of a particular cell, or if it's from human or mouse or whatever species. Be careful that when you do the digestion, it's not 100% digestion. It should be partial digestion. And I noted this in red because it's very, very important to retain this information. Because imagine this is a gene, okay? And you want to clone that gene. If you keep cutting it and you have 100% digestion, you probably don't have all the fragments covered. In fact, you want to have a partial digestion where one clone will have digested only here, so you get this part, and that is cloned to a vector. And then in another clone, this site is not going to cut, and therefore you get a bigger clone here, and that go to another vector. And then probably if you take this site and that site, well, this site here and that site, you can see that there's two sites didn't cut this guy here. And therefore, in this clone, you have a chance to clone all that into another vector. Before that important part of the gene and after that part of the gene, you can see that there is one restriction site here. These two restriction sites didn't cut, and then this one cut. So that's why you have to have a partial digestion, not a complete digestion. And this way you will get all chances of having these small fragments or these larger fragments. For example, between this guy and this guy, there were like 5 or 6 sites that didn't cut. This is very important because that is how the overlap happens. So between this clone and that one in this 3 green, you have a chance that it's overlapping there. So you would know that the sequence of this guy is followed by this guy. And then this one here is overlapping with that. So you would know that the sequence continue like this. And to get at the end, the sequence all over. So it's important to, we didn't discuss the sequencing part of the library. That's how the sequence will work. The sequencing will take this part and then it's overlapping with this guy. So we know that this sequence is exactly like this one. Okay, great. So we know the sequence still here. Now we move forward. We know that this guy is overlapping with this in this area. So we know now the sequence all the way here. Then this guy is overlapping with this. And that's how it goes. And we keep going and going. So each clone is adding more and more to our gene so that at the end of having all these clones, we will be able to generate a sequence that covers the whole genome. So this is the concept. This is exactly the concept. So how did we approach that? In the Human Genome Project, the idea was we have the DNA molecules of the human, and we want to know the sequence of all the chromosomes from the beginning to the end. This is a project that started in 1990s. It took 13 years to be covered. And it was run in 2 different companies, one that is a private and one that is public funded. So the first challenge we have is that the size of the DNA is big. It's like 3.2 billion base pairs. And then there's also 1% of the DNA that is very difficult to sequence because it's complex. It has GC-rich content, and it's part of the centromeres and the telomeres. And this is a part where we don't know much about the sequence. Also, when we do sequencing, you remember the practical aspect of the sequencing. There was a slide where I showed you that you can sequence DNA between 600 to maximum 1,000 base pairs. And after that, you start to get noise. So in principle, you can't have sequencing more than 1,000 base pairs fragments. So that's another issue that is added to our challenges. And in order to solve all these issues, this is the scheme that shows you how it works. So first, we will take all the DNA. Since it's very big, we have restriction enzymes. We're going to cut it partially, as I described in the previous slide. So you do fragmentation. So you get these fragments. And it's a partial digestion so that you still have an overlap. So this guy overlapped with here, overlapped here, and it's overlapped here. And then this guy overlapped there. So there's always overlap. And then you can jump from one to another and continue sequencing all the way. Then you can get the sequence from the A to Z. And all you need to do after that, once you do the sequencing, you will know the sequence. And all you need to do now is to put it like a puzzle back to give you the DNA sequence. So here is a YouTube video that explains the Human Genome Project. Some of the information on the slides are taken from that video too. So you can watch it and it really helps. So there were two approaches to obtain the human genome sequence. One approach, which is the very classical approach, that is called map-based sequencing. Recall that map-based sequencing, it also explains that you should know some information about the gene. And most probably you would know that this is a chromosome 1, let's say. And then you would use this process of golden tiling path. The golden tiling path is that you are taking this and you have a bridge between the two, and then you have another bridge between these two, and then again, and that's how you would know the sequence. And then you would need to perform two levels of cloning. The first cloning would be, I would take the DNA and then I will clone it into like 150,000 base pairs. It goes to bacterial artificial chromosome. And now this is one back, this is another back, this is a third back, and this is a fourth bacterial artificial chromosome, okay? Then I would refragment those again with restriction enzyme partially, as I explained in the previous slide. And then I clone it to a smaller plasmids, and I would sequence all these small fragments. Then I will rebuild this unit by rebuilding those guys here. And the same here, rebuilding those guys, it will give me this unit and the next and the next, and then I can create the sequence. Of course, this is a super accurate procedure. It takes a long time. However, its advantage is that it's very accurate. On the other side, it is the slide that I always explain to you from the beginning of the classes till now. And that's why, because it's much easier and it's more straightforward. You would take the genome and then you will simply digest the genome into different, with a restriction enzyme, 1 or 2 restriction enzymes. And then you will clone all these directly into plasmids. Instead of doing a back that is digested, that is then is sequenced and aligned, and then you can go back to the alignment of that, we directly go into smaller fragments that is subcloned into plasmids, and then we will realign those back. And this realignment needs a lot of powerful computational power and bioinformatics. Okay, so that's exactly how things done. Both projects start almost the same time and they ended at the same time. But nowadays, I think this would be much easier to follow. And that's why in most of my slides, I describe this type of library generation and genome sequencing. The advantage is that it's much faster and it's less expensive. The disadvantage is that sometimes it's prone to errors because you get some gaps that you cannot fill. So in the next couple of slides, there will be few more details on each of these two. Okay, so I'll make sure that slide is corrected. At least I can see it, but I will it's going to come more in the next slide again. So here I'm going in more details for you to explain how map-based sequencing or hierarchical shotgun sequencing. So you take first, sometimes you need information based on where is this location you are working at. And one of the steps that are usual in hierarchical sequencing is that you might probably sort first the chromosomes based on their size. So you take one chromosome per chromosome, and then you digest it. So you get these fragments, and each of these fragment clones are the one that go to the back. Okay. And then you would fragment those two. Like imagine you took this pack and you digested it with a restriction enzyme. It has 5 different restriction enzymes here. And then you start to get the overlap. You got it here, you got it there, you got it there. You get as many subclones as possible of this. Each of this subclone is going to a vector. So like this guy go to a vector, this guy to a vector, this guy go to a vector. And finally, you have one vector with a clone, 2, 3, 4, 5. So the sequence of the vector number 1, let me call this vector number 1, overlaps with vector number 2 or plasmid number 2. So if you do the sequencing, you know that this is the start, this is the end, and then you would know, oh, there's an overlap here. Okay, great. Then I would continue. I know the sequence here. Oh, there's an overlap with clone number 50. I don't know what's the number of that clone. Great. And then this clone overlaps with another clone here. And then this overlaps with this guy here. Now, when I map the sequences, it would tell me, oh, these 2 clones overlap here. These 2 clones overlap there. And these 2 clones overlap here. And therefore, the sequence is known from A to D. The gene A, B, C, and D are known. And when I know the sequence from the beginning to the end with no gaps, that's what I call a contig. So this is a contig that we know. It's a part of, it's a clone of the back. And that is the major block of the hierarchical shotgun sequencing. So take a plasma, chromosome, partial digest it, clone it to a back that is really big, around 150,000 base pairs, then take that particular back and do partial digestion of it and make subclones into vectors, and then you get the sequence of each of those as a contig, and therefore now probably you would know the sequence from 0 to 150,000 base pairs. So doing this many times, you have to cover in order to get one chromosome, and then you have to do for every single chromosome like that. And that's how the map-based sequencing worked. On the other hand, the whole genomic sequence, shotgun sequencing, is much simpler to understand, because as I said, you would take the genome, you would digest it into the smallest fragments possible, around 4,000 to 8,000 maximum. So you can put those directed into plasmids. And then you clone those plasmids into bacteria. So you transform bacteria. And then you can isolate those clones and you start sequencing. And bacteria number 1 or clone number 1 gives you that, oh, this is a fragment. And then bacteria number 2 or clone number 2 is going to give you an overlap. And then again, and more till you reach around 5,000 clones or so. And you need a very powerful algorithm to align the sequences and find the overlap. And it will give you then the sequence from A to Z. And then it will easily identify that chromosome number 1 start here and end there, because of course there's no continuity to chromosome number 2, and chromosome 2 the same, and all the end. And in fact, as I said, many times I described for you the library, the genomic library generation, and I was describing this easy one, which is the whole genome shotgun sequencing. Okay. Yeah, so these two slides we discussed many times. I just added here for you to remind you that we discussed this in two lectures. So, okay, now we already did the sequencing, but the question is, what information do we get and why are we doing this? And many of you are asking why he's teaching us this, I think, right now. Fair enough. Without knowing these sequences, we wouldn't be able to understand how the genes work, and we wouldn't be able to modify or manipulate any of these genes. So you could see that the accumulation of all those molecular biology techniques led to a stage where we can know all the genome. And now we know, if you ask any part of any chromosome, what is the genes before and after this location, you directly can go to NCBI or Ensemble, and you can see what are the genes in that location. Yet we have a 3.2 billion base pairs in our genome, but that doesn't reflect the biological complexity at all, because we have only less than 25,000 genes. And by the way, before the genome project has ended, people thought we have around 30 to 40,000 genes, and it ended up that we have much less genes than we expected. There are some amusing and, like, I want to say astonishing information here, that less than a third of the DNA is being transcribed to RNA. In terms of transcription, this means all the types of RNA, I'm saying, not only messenger RNA, meaning RNA, messenger RNA, ribosomal RNA, transfer RNA, long non-coding RNA, microRNA, all the families of RNA. So only 28% of the genome, so less than 1/3, is being transcribed. And only 1.25% of that is translated into proteins. And we know that the proteins are the functional units. So we are using around 2% capacity of our genome. The 98% of the genome that do not code directly for proteins, we still don't know all the function. And that's why the functional genomics is one of the biggest fields that is strongly recommended. And it will keep evolving as much as we are discovering new techniques and ways to study this domain. Around 50% of the, or a little bit more, of the genome is transposons, which are part of repetitive sequences, like small repetitive sequences like microsatellites and variable tandem repeats that we discussed in the paternity and the crime detection. Many, around like 5%, is duplicated in the genome, and we know that there are translocations that happen from one site to another in the genome. There will be an exam question for this red text here. Okay, now, astonishingly, like, we know that when there is a GC-rich area, it's difficult to study because the GC-rich area are difficult to have primers, and probably you saw that when you design your primers. But it happens that most of the GC-rich areas have genes. Probably it's a site of protection, and entrons on that areas of GC-rich are mostly shorter. So these are some of the information we got out of the Human Genome Project. Now, if you look for the complexity, as I said, the genome size doesn't really reflect the complexity. So if we look for a human, we have around 25,000 genes. If you look for rice, you have around 46,000 genes. So the number of genes doesn't reflect the complexity of the organism. Yet, there are still many things that we need to discover. But with the Human Genome Project, we were able to identify lots of sequences that could facilitate studying the genetic diseases and identify common and complex diseases like diabetes, asthma, cancer, and other type of genetic disorders. And by applying the information we get from the genomics, relating that into the transcriptomics, which is the RNA in the whole, the total pool of the RNA in a cell, then it helps us a lot to identify differences in terms of gene expression, as well as relate on the gene expression profiles. For example, what would happen if you use a drug? Does it change gene expression? Does it elevate a gene expression? Does it block a gene expression? Or you can link between the transcriptomics and the genomics, how pathological states can affect a certain aspect. Let's take an example in conditions of heart disease. A mutation can cause a heart disease, and therefore you could detect the mutation at the DNA level without really studying the transcriptomics and the proteomics. You wouldn't really know what is the cause. But now, having the sequence, then knowing the type of the transcriptome that we get in terms of a mutation and what type of protein we get, then we can relate that to the pathological state. And if you look here, I don't expect you to know these, but it's important for you to really keep in mind that the majority of the genes in our genome, we don't know the function. And if you look number 17, it is molecular function of these genes are not known. It's almost 45% of our genome. So this is a domain that is really understudied, and with the technologies we have right now on bioinformatics and the sequencing tools, we will have much more information in the future. So this slide I'm visiting again, just to remind you that, as I said, we know that now the Genome Project is there. We know the sequences. Excuse me. But knowing the DNA sequence itself only is not enough. So we have to relate that to other aspects or other processes, such as transcription, or in other words, the messenger RNA or the total amount of RNA that is being transcribed from the given genetic material. And we have to think about the complexity that comes on top of this by splicing the RNA. So you remember we have a gene that is generated by transcribing the DNA into exons and introns, and then we have to splice that out. We have to remove the introns, and we keep only the exons here, and that's what we call splicing. Then we have the translation process, where this messenger RNA is most of the time, as a functional gene, it will be translated into a protein, and that protein will perform the function. For example, people who are having diabetes, they lose the expression of the protein insulin. Therefore, at any of these three levels, if anything affected the gene that produces insulin, if it's not transcribed correctly, or if it's not transcribed into RNA correctly, or it's not being spliced and translated to a protein, all that leads to the loss of the insulin. And therefore, the function of the gene can be studied at all these three levels in order to recompensate or recuperate the function. Nowadays, what we do for people who don't have insulin, we simply inject them with the protein, right? We produce a protein and we inject it in their blood. But at this point, there are some diseases, and we will discuss some in the future, that we could really play at the level of the messenger RNA and the level of the gene. Okay, remember that RNA splicing is very important for protein generation for two reasons. I will describe this in the next slide, but also there are some stabilizing factors for the messenger RNA, like adding the poly-A tail and the cap at the G's at the 5' end. So, and I remember I asked you a question about this in the exam. So then, where does the biological complexity come from? It comes from, first, the alternative splicing, and I'll go back to the slide, because if you take this exon and this exon and this exon, you can make a protein that is different from if you take this exon and that exon. Okay? So the alternative splicing adds a lot of complexity to the transcribed messenger RNA, and therefore a different protein can be generated. On average, and you could see in the assignment that in your assignment, probably there were 7 transcripts from the same gene. On average, there's at least 3 different messenger RNA that come from a gene. So therefore, if we have almost 24,000 genes, if you multiply this by 3, you could expect around 72,000. This is not an accurate number, but this is an approximation. Also, for the proteins that are being generated, there are post-translational modifications. Some of those would be adding chemicals to the proteins, and therefore the protein can be acetylated, so an acetyl group can be added to the protein, or a phosphorylation where a phosphate group can be added to certain amino acids. So that's another level of complexity. And the third level of complexity is going to be protein-protein interaction, where protein A binds to protein B to make the function better or to block the function. So to activate or inactivate a function. Or go to the nucleus, shuttle to the cytoplasm, go back to the nucleus. Send a signal. Like all this protein-protein interaction, it's going to come in the future. But again, this slide is very, very important. And this slide will be included in your exam. Okay, I still have a few minutes. I'm not going to go to the proteomics today. I'm just going to describe a little bit about the transcriptome. So studying the genomics is mostly knowing the sequences, the locations, the neighboring gene, the upstream sequence, the downstream sequence. Studying the functional aspect or the functional genomics, it will characterize not only the sequence, it will characterize the function by which that sequence will perform. And therefore, it will look at the 2 downstream levels that I was just describing you here, which is the transcriptome and the proteome, which is all the proteins. Therefore, it's here, it's saying the functional genomics, it includes the identification of all the RNA molecules transcribed by the gene or the genome, and that's the transcriptome. And it will also study the function of all the proteins encoded by a genome, and that's what we call the proteome. But these 2 fields were not possible before having a really, really strong bioinformatical tools that should side by side go by the laboratory experimental procedures and approaches to identify a gene and then guess what that gene will do and then validate that. So proteomics and transcriptomics need very good skills in bioinformatics as well, and very good tools and algorithms. So if I would like to study the transcriptome, here's the roadmap. So let's say I want to study or the messenger RNA in a cell. You have cells in culture. So you would go and isolate the RNA. We know how to isolate RNA now because we described this in the previous lecture. Whatever method you use, you could isolate all the RNA. And then there is one very important characteristic of messenger RNA. They have the poly A tail that is added. That is not added to other RNA. So then you can use the poly A tail to generate by reverse transcription using a primer that is poly DT, because you have A at the end. You can use TTTT as a primer to generate, with the help of the reverse transcriptase enzyme, a complementary strand, and that's why it's complementary DNA, cDNA. Then you need, obviously, the cDNA now, it's hybrid, one part RNA and one part DNA. You would need to generate the second strand to make a complementary double-stranded DNA. And at this point, you are more or less similar to having a genomic library, because you generated all messenger RNAs and you make all those messenger RNAs double-stranded DNAs at this point. And all you need to do is just clone all those into a library, exact same way we used by restriction digestion, and then make recombinant vectors, put those plasmids into bacteria, and now you have a library of all the cDNA. Of course, there is one question here that I didn't cover for you because it's going to be a little bit more complicated, is how do we do the cloning step, because now we cannot digest. So we have to make sure when we do the primer, probably I have to add this in the next lecture, a slide just to describe this. When we add the primers, we add some adapters with specific restriction enzymes, so that when we reach this step here, we can use only that restriction enzyme to clone the DNA into the vectors, and then we get the library. So how does it look like in terms of steps? So this slide is just reassuring what I just said. So the cDNA libraries are generated from the pool of messenger RNA. Because the messenger RNA is not stable, we need to generate a complementary cDNA by reverse transcription. And this is an enzyme that we got from RNA-containing retroviruses. We described this before. Then in order to generate the cDNA library, we can study multiple things. After generating the cDNA library, we can study multiple things, including how to modify a protein. So you can change something in a protein at the level of messenger RNA, and you can study how it works. Or you can also study the specificity, the tissue-specific expression. In other words, the messenger RNA of insulin probably is not generated except in pancreas. So the steps are, you isolate the RNA, you synthesize the complementary cDNA, then, of course, the primers should contain the adapters, then you digest the cDNA borders, and then you clone those into a vector that is digested with the same enzyme. So you got it here. You make a cDNA, you make it complementary double-stranded, you digest the borders, as you can see, and you take another vector, which is a plasmid here, you got it with the same enzyme, you make a vector. Each of these go there to make a clone, you transform the bacteria, and now you have a library of cDNA, which comes from the messenger RNA. And this way you can study the transcriptome. Nowadays, we don't need to do all this. Nowadays, we have many other tools to study the transcriptome. There are three major ways. In this, you can see what we call microarrays. There will be a lecture where we describe the RNA sequencing and the RNA sequencing in single cells. So it's called single cell RNA-seq. For some reason, this flopped. It's called single cell RNA-seq analysis. This should be here, okay? Now, few words about microarray, and I will revisit in the next slide, in the next lecture. With microarrays, and I will jump to the next because it's much easier. We have a question here that we would like to understand what is the difference between a cancerous cell and a non-cancerous cell. So simply you can take these two cells and you can take RNA, messenger RNA, let's say, and then you can generate the cDNA, but also you can label that cDNA to make it different. And then you can hybridize, make these RNA, double-stranded DNA hybridize to certain units or flow cells. And after that, you simply can look at the expression level of the specific genes by looking into the color. Each of those dots is a gene. And the green means that the non-cancer cell expresses more of that gene, and the red means that this particular gene is more expressed in the cancer. Like with this approach, I can identify what are the genes that are highly expressed in a cancer, because the more this cancer cell will produce RNA, the more the gene is active and the more transcription is taking place. And therefore, the more of the green or of the red fluorescent you will get of that gene, it will tell me that this gene is active. The same thing, if you want to see the downregulation, you can look at this at another level. You can say, oh, in wild type or non-cancerous cells, they have much more of this green, but much less of that red. Using this technique, there were like almost 70 genes that were expressed that we know that they play an important role in cancer. So this is one of the approaches. Again, because the time is really short, I'm not gonna describe this again and again now, but in the next lecture, I will revisit it, but it will come also as an introduction for the RNA sequencing. Now, in the next lecture, because we will just close everything about DNA, I will describe for you the genetic manipulation by making transgenic animals, and this will be the last lecture about the DNA in this class. Okay, I'll stop here. If you have any questions, please stay. And you can unmute yourself or write in the on the chat. Thanks, Maggie. Any questions? Anybody has questions? All good. Thanks, Malik.
Sorry, Professor, I have a question.
Go ahead. You have a lot of noise, Kenan.
Sorry?
You have a lot of noise from the, I think it's echo coming from your mic.
Okay, let me try to type it in the chat if that's better.
Oh, yes, please. Thank you. Okay. Okay. When you okay, Gary, you're asking when you're getting a fragment that overlap, are you using one restriction enzyme or running it many times? So you generate the fragments by using one of the restriction enzymes. So the fact that you got the first round of digestion, Gary, then when you have this first round of digestion, it is partial. So in other words, let's say you use echo R1, only echo R1. Echo R1 is going to generate, let's say, 6,000 different fragments of the whole genome, potentially. If you add echo R1 for 3 hours, it might digest the fragments 100%. If you add it 1 hour, it will have, let's say, 20% possibility. So you have to do it a partial digestion, either by minimizing the time or minimizing the enzyme. So this is a step I didn't really explain to you too much because you will never be doing this. So then, because the digestion is not complete, that is what creates the overlapping. Because one side sometimes doesn't cut. Normally, if it cuts 100%, you will get only one fragment. But since one of the enzyme sites didn't cut because that's a partial digestion, then those fragments are generated. And the more fragments you generate, the more chances that you will get overlap. Did this answer your question?
Yes, pretty much. Thank you.
Yeah. So you're assuming many different. No, I mean, you don't want to do multiple restriction enzymes because one is going to be enough. And one is going to be much easier to manipulate. So the other issue that we didn't describe is that if you use 2 restriction enzymes, for example, some of them are not going to be specific 100%. So they can cut somewhere else. You don't want that to happen. So that's why 1 restriction enzyme is enough, but you don't want to cut 100%. So you either minimize the amount of the enzyme you use for the DNA or you minimize the time so that some sites cut, sometimes some not, and then the overlap will be generated because many fragments will be generated. Some of the fragments are containing different. Now, Gary, one concept, your question is very important because we don't have only one copy. You have many, many copies of the DNA. That's something you have to remember. When you take a group of cells, you have a group of DNA. So the same DNA can be cut in different patterns. It's not only we have one particular only sequence we are cutting. We have many, many of that same sequence. Okay?
Yeah, it's very clear now. Thank you.
Okay. Any other questions, guys? If not, I still see 4 people here. If anyone has any questions, please tell me. Okay. Thank you very much. Have a good afternoon.